Welcome to the Comparative Cell Membrane Lab. Now, essentially what this lab is going to be doing is focusing on the concept of simple diffusion. And remember, simple diffusion is molecules moving from an area of high concentration to low concentration because of Brownian motion, because those molecules are constantly wiggling and jiggling and wiggling and jiggling. The other thing that we're going to be working on with this lab is we're going to be working with the concept of osmosis. Now, osmosis, remember, refers to water moving, and water will move for two reasons. Water is a molecule, so it will move, according to simple diffusion, from an area of high concentration to low concentration. But water is also a solvent. It wants to be where the stuff is. It wants to be where the solute is at. So water will move from an area of low solute to an area of high solute. It's attracted to the stuff. It's attracted to the solute. It wants to be where the solute and stuff is at. So those are two reasons why water will move. And so one of the cool things that we'll be doing with this lab is we're going to be able to measure and see those molecules moving. The other thing that we're going to be doing is we are going to be modeling a cell. We're going to be modeling the plasma membrane, the cell membrane, which is semi-permeable, allowing certain things to come and go. And we're going to be able to see the changes. Now, this is really nice because a lot of times in cellular biology, we don't get to see things changing. So that's really the emphasis or the focus of this type of a lab. Now, one of the things that happens whenever we're dealing with simple diffusion or osmosis, molecules need time to be able to move. And so one of the things we need to do is set it and forget it. And we do this a lot in science. It's not unusual for scientists to set up an experiment, go home, come back the next day, and then read the results. Now, for this experiment, we don't have to wait quite that long, but one of the things that we will be doing is, for experiment 1, we'll set up a sugar solution, we'll get the experiment started, we'll let it sit for 24 hours, and then the next day we'll come in and do a very quick measurement on what might have happened. So please note that with this lab, you are going to have a little bit of a duration. So keep that in mind. You're going to set things up, kind of forget about them for a little while, but don't forget too long, and then continue on with your experiment. So I have the setup of supplies, and one of the things that I wanted to point out is in this week's lab, we are going to be testing for sugars again. So in your comparative cell transport packet, you'll notice that here we have our Benedict's reagent again, that blue reagent testing for monosaccharides, simple sugars. And I decided to go ahead and pull out my fuel and beaker again. I'm going to use that same method that I used with the biological molecules lab in order to heat up my Benedict's reagent and get that reaction to occur. So what you see here is a little bit different than what is being asked for in the hands-on lab material list, and that is intentional. So once you have all of your supplies, the first thing that we need to do for experiment 1 is we need to set up our sugar solution. Once we have our sugar solution and we make our sugar solution into different dilutions, in other words, different concentrations of sugar in different test tubes, then we're going to be cutting what essentially look like little French fries, little bits of white potato. And we want to make sure that we don't get any skin when we cut our little French fries. We want that nice solid inside of our potato, and then we will be putting our potato into the treatment and then reading the results in the next day. So for experiment 1, when you cut apart your potato pieces, one of the things to keep in mind is once you cut apart one potato piece, you may want to use that as a template. So you can cut all of yours. We need 2 potato French fries per tube. You can see I've got my 6 tubes all lined up here, all labeled A, B, C, D, E, F. I've added my different solution amounts, and I've added my 2 little potato chunks. So here's my last test tube that I need to add my little French fried potatoes to. And one of the things that I love about this type of experiment and any type of experiment is that we have a double check. We've got this visual double check to make sure that we did everything accurately. So when I look across these test tubes, what I can see is that the volume in every test tube is the same. Even after adding my potato chunks, all of my test tubes have the same amount of liquid. So that means that not only did I fill my test tubes up with the right amount, or at least the right volume of liquid, but my potato chunks also have a lot of consistency. That consistency is going to help me to determine what's going on in my experiment. Remember, whenever we set up an experiment, we need to know that our dependent variable, in this case, the weight of our potato, at the end of the incubation period, we need to know that that is due to what we changed and what we manipulated and not due to anything else. So, we've got the same type of test tube. I used the same potato for all of my little French fries. I used the same sugar and mixed up the sugar concentration in the same beaker. I used the same pipette for distilled water and the same pipette for my sugar solution when measuring everything. I made sure that all the volumes in all of my test tubes were the same and that all of my little French fries were all the same. I held everything constant. So my independent variable, my concentration in sugar, is the only thing that I have changed in every single test tube. And that should influence my data, my dependent variable, at the end of experiment 1. Now for experiment 2, as I mentioned before, we are going to be making a model cell. And in order to do this, we're going to use the dialysis tubing. Now this is the dialysis tubing that they use for people who are whose kidneys are struggling and starting to fail. And this is essentially a semi-permeable membrane. It's going to let certain things go through. And when you first unfold it, you're going to notice it's super crinkly. And that's why the first thing we need to do is to get a cup full of distilled water. We dump in our dialysis tubing, and this is going to start to soften that up a little bit more. Make it a little bit more malleable so that we can experiment with it. So, my dialysis tubing is soaking in my distilled water. In my graduated cylinder, I've already measured in the required amount of distilled water. So I'm going to add in my starch solution. I'm going to give it a little bit of a shake in case any of the contents have settled. And for my starch solution, I know that I need 2 total milliliters. So I'm going to drop very carefully and keep an eye on it. So I've added my starch. The next thing I'm going to do is add my glucose solution. Again, another 2 milliliters. My total volume in this graduated cylinder should be 8. And I know that what I need to do is get all of these compounds thoroughly mixed up. So I'm going to put my thumb over the top and give this a little bit of a shake. Start getting this really mixed up. I'm going to dump this into container 2. If I wanted to, I could take that stir rod and stir it around again. Just want to make sure that these contents are really well mixed up. Hopefully by now, my dialysis tubing is soft enough to work with. So the trick here is to not let it dry out. We want to move relatively quickly. So on one end, I'm going to give a couple good folds, and I'm going to take my cut rubber band, and I'm going to use this and tie a super tight knot here. We need this really tight. We do not want our cell to leak. So I captured the fold here with my cut rubber band. I dipped the dialysis tubing in again because I know that this tends to dry out so quickly. If yours does dry out, that's no big deal. Just put it into the cup of water. I'm now wiggling my tube. This gets the tube to separate. I'm going to test it for any leaks. The way that I'm going to do this is I'm going to take my little pipette and my distilled water. I'll put a couple milliliters into my model cell. And I can already see I got a little bit of a leak. I want zero, no leaks. So I'm going to dump out my water and tie that rubber band a little bit tighter, put a second loop in there. I want to make sure that my model cell does not leak. Test again. And just because I want to make absolutely positively sure, I'm going to dry this off. Grab a new paper towel. Set it on that new paper towel. Kind of push around a little. And we have no leaks. Excellent. Going to dump out that distilled water. That was just to make sure my model cell doesn't leak. My dialysis tubing is a little bit dry, so I'm going to wet it again. Wiggle it, make sure it's nice and loose. I'm going to take my funnel, slide it in to my dialysis tubing, and then very carefully I'm going to pour the contents of cup 2 into my model cell. There we go. Set this off to the side. Squish all the air out of your model cell. Give 2 big generous folds. We want to leave a little bit of floppy space in here. We're going to tie it with a rubber band on the other side. Make sure we got a really good seal. And then the next step will be to add the iodine to cup number 1. There we go. We are going to add 20 drops of iodine to cup number 1. Once you've added your 20 drops of iodine to cup 1, give it a really good stir. We want to make sure this is a nice homogeneous mixture. Now, one of the things that we're going to be looking for is we are going to be looking for color change. So right away, I'm going to set up my experiment with a white paper towel beneath it. I've got my cup of iodine ready to go. I've got my model cell all dried off. I'm going to drop my model cell into my cup of iodine. I'll be watching for color change to happen over the next hour. That's why I set it on top of a white background. One thing I will point out to you is that if your model cell leaks, you are going to see a color change outside of your model cell, and that's not expected. What we want to have happen is for things to potentially change inside of our cell. We do not want anything to change on the outside. When you are done with this experiment, you're going to be opening up your model cell and dumping the contents back into cup 2 to test, and that's where our Benedict's reagent, and in my case, the beaker and the flame is going to be important. Remember, I'm just using this to heat up my test tube with Benedict's to get this reagent to change and show me if I have any monosaccharides in my test tube. So follow those end protocols to get the end results and the end data. Hopefully this result and experiment works well for you. Remember, when we're dealing with osmosis, when we're dealing with simple diffusion, and we're waiting for molecules to move, we need to set it up, put it in a place where it won't be disturbed, and then come back and make our observation later. Please email me if you have any questions.